Hello students, in our today's session we will see about the genetic code and the characteristics of genetic code, which is covered in the chapter Molecular Basis of Inheritance. Students, it is already known that DNA is a master molecule of a cell that initiates, guides, regulates and controls the process of protein synthesis. To perform this complicated function, it must carry the required information for the synthesis of proteins. Obviously, this information has to be located in the DNA itself. The site for storing this information lies in the sequence of the nucleotide. We have about 20 different types of amino acids that are involved in the process of synthesis of proteins. DNA molecule has 4 types of nitrogen bases to identify these 20 different types of amino acids. So the question arises then, how is it possible that 20 types of amino acids are encoded by 4 types of nitrogenous bases? According to Crick, the information on the DNA is stored in the form of coded language, which is called as a cryptogram, also called as the genetic code, that contains code words, codons, each one specifying, that is representing a specific amino acid. Genetic code therefore is a collection of base sequences that correspond to each amino acid. A single nitrogen base in a codon, that is a singlet codon, will encode for only 4 different types of amino acids. A combination of 2 nitrogenous bases, a doublet codon, will specify only 16 different types of amino acids. A combination of 3 nitrogenous bases, triplet codon, will specify 64 different types of amino acids. Therefore, George Gamow suggested that in a codon there must be a combination of 3 conjugative nitrogenous bases that will be sufficient to specify 20 different types of amino acids. So you have 3 bases at a time which code for a particular amino acid. Students, let us have a look at the experimental evidences for proving genetic code is a triplet code. The first evidence for genetic code to be a triplet code was given by Crick using the frame shift mutation method. Now there are a number of types of mutations that happen. We will just concentrate on the frame shift mutation. This is the mRNA and 3 nucleotide sequences make one codon. So you read one codon at a time and each codon codes for a specific amino acids. So when you're reading 3 at a time, if you skip a particular alphabet here, instead of A, you shift the frame and you read from C, and you give, instead of having a GCA, you read it as GCC, and since you have missed one, the reading frame shifts and it changes the rest of the codons, and because of that the amino acids that are supposed to come together for the formation of proteins change. So this gives rise to a new protein, and this kind of mutation is called as frame shift mutation. This thus proved that 3 conjugative bases form one triplet codon. The next experimental evidence was given by Nuremberg and Matty. They were able to synthesize artificial mRNA which contained only one type of nitrogenous bases, example uracil, which is also called as a homopolymer. They synthesized poly-U sequences which they transferred to protein synthesizing enzymes. A small polypeptide molecule was produced, formed by the linking of phenylalanine molecule. Here they used only one nucleotide, that is, example is they formed UUU, CCC, AAA. They are poly-U contained codons, triplets of only uracil, and they found out that 3 U codes for phenylalanine. Later different homopolymer codes were also found out. Codons formed by 2 or more bases were also tried. Then we have Dr. Hargobind Khurana, who devised a technique for artificially synthesizing mRNA with repeated sequences of known nucleotide. He used synthetic DNA and he prepared chains of polyribonucleotides with known repeated sequences of 2 or 3 nucleotides. For example, CUC, UCC, and again a repeat of CUC and UCC. This resulted in formation of polypeptide chain having 2 different amino acids placed alternatively, which were leucine and serine. Similarly, polynucleotide chains with 3 nitrogenous base repeats gave polypeptide chains with only 1 amino acids. Later, we have Servo Oak established that the enzyme polynucleotide phosphorylase was also helpful in polymerizing RNA with defined sequences in a template-independent manner, that is enzymatic synthesis of RNA. So these people worked together to find out and conclude that the genetic code is a triplet code, and all these individuals received a Nobel Prize for the same. Finally, students, we had Nuremberg, Matty, and Oko depicting all the 64 codons in the dictionary and forming this chart showing all possible nucleotide sequences or the codons that code for a particular amino acid. Students, let's have a look at the characteristics of genetic code. The genetic code is a triplet code. This is what we are looking at. 3 nucleotides together form, or the 3 bases together form a triplet code, and the code is always read in 5-3- direction. In every living organisms, this code is a triplet code. Now the code has a distinct polarity. It is always read in 5-3- direction and not in 3-5- directions. Otherwise, the message will change. For example, at the 5-3- direction, the first thing that comes at the 5- end is AUG. The genetic code is non-overlapping. Codon is non-overlapping. Each single base is part of only one codon. Have a look here. The first codon is CAT. Then if you have to see an overlapping, the T may get repeated again, TGA. Or you have a second way of overlapping where the after the first 3 nucleotides in the codon, the next 2 are again repeated, that is ATG. So such overlapping does not occur. You only have the nucleotide or the base participating once only in a codon. The genetic code is commaless. You do not have any gap or a punctuation marks. So you read 3 bases at a time in a continuous frame. You can't miss out on one, or neither can you overlap any single base. The genetic code has degeneracy. Usually, single amino acid is encoded by single codon. However, some amino acids are encoded by more than 1 codons. Cysteine has 2 codons, while isoleucine has 3 codons. This is called as degeneracy of codons. Degeneracy of codon is also explained by Wobble hypothesis. So if you look at the chart, you will see that more than 1 codon codes for a particular amino acids. Students, in this picture, you can clearly see the phenomenon of degeneracy of the genetic code. That is, here you have 2 codons coding for 1 amino acid. Here you have 4 codons coding for leucine. Again, here 4 codons for valine. Now, this phenomenon is explained with the help of Wobble hypothesis. I take up all these codons here which code for valine, and you have your transfer RNA form which has the anticodons which base pair with the codon on your mRNA. So if you see in the base pairing, here you have all a perfect codon anticodon pairing, but in the other cases of codon, the last pair of the bases is not showing a perfect pairing. Only the first 2 bases are showing a perfect pairing. So such base pairings are called as Wobble pairings, and these are called as a Wobble base pairings. But in all this case, the same amino acid is delivered with these 4 different types of codon, that is valine. This phenomenon is called as Wobble hypothesis. Students, the genetic code is universal. By and large, in all living organisms, the specific codon specifies the same amino acid. For example, codon AUG always specifies the amino acid methionine in all organisms from bacteria up to humans. The genetic code is non-ambiguous. Specific amino acid is encoded by a particular codon. Alternatively, two different amino acids will never be encoded by the same codon, which means that one codon will never code for more than one amino acid. We have initiation codons and termination codon. AUG is always the initiation codon in every mRNA. An image of the mRNA, you will see at the 5' end, you have AUG, which is the initiation codon, and it codes for the amino acid methionine. Out of the 64 codons, 3 codons, which is UAA, UAG, and UGA, these are termination codons which terminate or stop the process of elongation of the polypeptide chain, as they do not code for any amino acids. Here you have these codons that are there which are coding for specific amino acids, and as the process of translation occurs, each codon will bring in a new amino acid and form the protein chain till a point it reaches the 7th codon here, which is UAA. It is called as the stop codon. So if you see any one of these 3 codons at the end of your mRNA, that is at your 3' end, it will stop your protein chain synthesis because they don't code for any amino acids. So then I'll get a primary protein structure with just 3 amino acids forming the polypeptide chain. The genetic code is universal. Usually in all organisms, the specific codon specifies the same amino acid. Codon and anticodon. Codon is a part of your DNA. Example, AUG is a codon. Now this AUG is a codon which is a part of your DNA, and it is now transcribed to your mRNA. It is represented as 5' AUG 3'. An anticodon is a part of your transfer RNA which is represented as 3' UAC 5'. You will see that they come together as complementary base pairings. A pairs with U because in case of your RNAs, the thymine is replaced with uracil. Rest, it follows the same rule of the complementary base pairing. So students, these are all the characteristics of the genetic code. I hope you have understood all the characteristics. Thank you students for participating in this lecture. We will meet soon again with the next topic. Till then, take care, be safe and keep learning.